such, we're going to go through DNA replication, transcription, translation, all of which you've already covered in general biology, so it should be a refresher from that. What? Huh? 7 years ago? We don't talk about how old we are. We talk about how young we are. All right. But in any case, though, that should be a refresher. We'll cover mutations. You covered that in general biology. Again, should be a refresher. What we will be doing differently, though, is there's some microbial genetic control mechanisms in place, transcriptional controls, and that'll be new for sure, as well as horizontal gene transfer. That will be new. Okay, so those items are where we're going to see some differences where you might it's like new material definitely. So this is a pretty big module, has a lot of information in it. So the general learning objectives that we have are just listed. So structure of DNA and RNA, again, refresher, recognizing the steps of DNA replication, that should be a refresher, that idea, the leading strand and the lagging strand of replication, the steps in gene expression as in transcription, translation, right? We're going to be comparing, of course, transcription to DNA replication, the different roles of RNA, messenger RNA, transfer RNA that play in transcription. We have the, going into translation, we'll go through that process. This definitely will be new, regulation of inducible and repressible operons. Probably won't get to that until at earliest this Wednesday, maybe on Monday. And then distinguish between different types of mutations, so that should be a refresher hopefully. And then this will be definitely, so 8 and 10 are the two that are going to probably be completely new information for you guys. And that is distinguish conjugation, transformation, and transduction. The rest of it, hopefully, you'll remember a little bit from your general biology, as well as to some extent, that module 2, when we talk about the chemistry of the macromolecules, there'll be a little bit of a refresher from that too. So to start off with, distinguish the structure of DNA and RNA, we have the nucleotide. The nucleotide consists of course, our pentose sugar, the phosphate, and the nitrogenous base. The pentose sugar, if it's a deoxyribose, then that is for what we're going to see in DNA. The ribose sugar is what we're going to see in RNA. And really the position at our sugars, they usually give the carbons a number designation in that carbon skeleton. Not something you have to know how to do yourself, so you're only going to have to know the ones that we actually talk about. And this is the pentose sugar in the nucleotides is one, and so DNA and RNA is one of those that you just get familiar with. And in the second carbon for a ribose sugar, there's a hydroxyl there. Okay, in the deoxyribose sugar, in that second carbon, it does not have the oxygen, deoxygen, right, without the oxygen. So that is the position that differs between these two. The two carbons that are key, though, for the process of making that nucleic acid is the 3' carbon with the hydroxyl that it sits there, and then the 5' hydroxyl, the 5' carbon where we see the attachment of the phosphate group. Okay. I'll continue with our backbone once we get to making a nucleic acid. But as a reminder for our nitrogenous bases, for DNA, the nitrogenous bases are cytosine, thymine, adenine, and guanine. Guanine and adenine are your purines, and again, the phrase would be pure as gold. That's probably the easiest way to remember those. And the pyrimidines are cytosine and thymine. The pyrimidines, though, in the case of RNA, you don't see thymine, you see uracil instead. Okay. So sometimes you will see actually all three listed together, right? Cytosine, thymine, and uracil, which of course pyrimidines makes you think of pyramid, right? Should have, think my 3 points, my 3 sides, triangle, right? So we have 3 in that group. The pyrimidines have a single ringed structure versus the purines have a double ringed structure. Now these nucleotides will link together to give us, give me 1 second, make sure I didn't link it yet. Link together, of course, to give us our polymers, the nucleic acids. And there is a difference between the two. DNA in the cell is found as double-stranded, okay? And what we see is the sugar phosphate backbone is the constant in that structure, and the nucleic acid, the nitrogenous base is what is going to give that strand its unique sequence, so the order of those nitrogenous bases. That order is actually going to be important in the interaction that it exists when we are using these strands of either DNA or RNA as a template, okay? DNA is found double-stranded, RNA in the cell is found generally single-stranded. That being said, single-stranded RNA does have the ability to base pair with itself at different locations, so we can see the interaction between base pairs of the same strand. This does then give some of our RNA molecules 3D structure. Okay, that additional level of 3D structure. The interaction between the base pairing that does occur. Do I have my slides out? Okay. Importance of this. I'll get to the interactions of those hydrogen bonding here, the base pairing in a moment. The RNA molecule that does base pair, one of the structures that we do definitely see this occur at is in this case, transfer RNA, okay, where we're seeing an interaction between the base pairing on the same strand of DNA, creating what we refer to as a hairpin loop. In this particular, in the tRNA structure, that this hairpin loop gives us almost, if you were to lie it flat, like smash it down flat, it would give kind of a 3-leaf clover sort of appearance. Of course, that's not how it truly appears when it with that 3D structure. It looks more along the lines of what you see on the right-hand side. So both the DNA and RNA can have additional interactions with base pairing that can occur, even though DNA is recognized specifically as always being double-stranded, right? And that's the natural state within the cell. RNA is naturally found as single-stranded. RNA can have base pairing also. Okay. Now, when we are making a strand of nucleic acids, so that making that polymer there to produce. In this case, you can see we have not identified specifically by our number 2, right? Carbon as 2, whether we're talking about DNA. And honestly, in this case, because we have the thymine, we can see it is DNA, but the process is going to be the same regardless of whether it is DNA or RNA. When we are making the polymer, when these nucleotides are being joined together, we have a 5' phosphate, okay, and a 3' hydroxyl. The 3' hydroxyl is what's going to interact with the 5' phosphate of the next nucleotide being added. So when we are making a nucleotide, it is going to be made in the 5' to 3' direction. okay? And that has to do too with the enzymes that are going to be involved in this process. It's always 5' to 3'. That whether we're talking about whether we're talking about DNA replication and making a complementary strand to a template DNA, or transcription, making a complementary RNA strand to a DNA template, or even when we're talking about translation, when we are having the ribosome read a messenger RNA as a template to make protein, it is going 5' to 3'. Okay. So in this strand here, the growing strand would have a nucleotide added to this 3' hydroxyl. Okay. The bond that forms is the phosphodiester bond, and that is what you see formed right here. Okay. The phosphodiester bond is what is the bond that we recognize as linking the nucleotides together. If we are looking at this structure, this here becomes our backbone, the constant. It is a sugar, phosphate, sugar, phosphate, sugar, phosphate, etc. backbone. So it just alternates back and forth between sugar and phosphate as you move up that backbone. What differs is, again, the nitrogenous bases. That the sequence of those nitrogenous bases is what makes that particular molecule unique. That is also then what confers the information that we find in DNA. All right. Oh, and also to note, because we will be bringing up the use of DNA at various points, even in the lab. Do note, of course, the phosphate has what charge? Negative charge. So both DNA and RNA have a negative charge to them. Okay, that'll come into play if you took general biology here when you did electrophoresis. We'll do that later in the term with the lab. Electrophoresis is you're going to be using electric current, and so we want to make sure we set up the current so our negatively charged DNA is going to move towards the positively charged, right, the cathode. Okay, because of the fact that there is that charge attraction. All right. The interaction between the two strands of DNA is going to be create this double helix formation. The double helix is one in which we have base pairing occurring with the nitrogenous bases. The base pairing is due to hydrogen bonds. There are 3 hydrogen bonds that will interact between guanine and cytosine, and 2 hydrogen bonds between thymine and adenine. Okay, you say, why would that be important? Well, it is important because it takes more energy to break 3 bonds, hydrogen bonds, than it does to break 2 hydrogen bonds. So during, say, at the front of a gene, for instance, we have a promoter. So we want to promote the separation of the DNA strands in order for us to be able to create through transcription, create a template of messenger RNA. Well, we're going to tend to see in the promoter region a area that is high in is AT rich, high in AT interactions, right? Which would make it easier for breaking apart those 2 strands. Also, as we read through Module 6, if you went through the text portion of it, and I made mention to it, but they do talk about it in regards to, I think it was either in the pH or the temperature one, on the fact that when you're looking at, say, higher, I think it was higher temperature, they're going to tend to have more in their genetic material. They tend to have more GC present because it's harder to break. It's just more, right? More hydrogen bonds means more energy to break that interaction. Okay. What happens when you do separate out break these hydrogen bonds? What is that process called? What is it called? We do that to protein.
Isn't that
Denaturing.
Denaturing protein. Well, you can denature DNA also. DNA has, so a lot of times we focus with the temperature and the pH, we're always focused on enzyme activity, right? Enzyme activity drops because of the fact that you are breaking the bonds, those hydrogen bonds, ionic bonds, etc., the 3D structure of the proteins. And yes, that is true, but it is also going to be true that you will be breaking the hydrogen bonds between double-stranded DNA too, if temperatures are high enough, and that will affect the ability and survival rate of that particular cell also. Okay, so it too is subject to denaturation. All right. Let's see, I don't want to talk about the major groove, minor groove. We're not going to get into all that today. Let's see. Did I talk about anti-parallel yet? I did in my head. All right, I was probably going and then got sidetracked and moving forward without. All right, so looking at our two, here we have our double helix, so two strands interacting together through that base pairing. So it is a double helix, but if we were to flatten it out, right, it's going to look something like this. And you'll notice, so this is one strand, this is the other strand. You'll notice that these two strands are what we refer to as anti-parallel. Okay, anti-parallel has to do with the backbone portion, the sugar phosphate portion. On one strand, we see the 5' end, right? It's going to match with the 3' of the other strand, and vice versa. Okay, that is referred to as anti-parallel. Of course, will come into play, of course, when we're talking about DNA replication and transcription, and when we're trying to use this as our template, because it'll tell us which direction we need to go in this process of polymerizing a new strand of DNA or polymerizing a messenger RNA. Now, the DNA molecule, because of the unique sequence of that, those nitrogenous bases, the DNA is going to be how we transfer genetic information from parent cell to offspring cells, so to daughter cells. Okay, so that is the direct way of what we call vertical gene transfer. Okay, vertical would be parent to daughter cells. Okay, all of the genetic information that's found in the cell is composed of DNA. I don't know if did we go through that in this class already? The sources of the genome for prokaryotic cells and eukaryotic cells? Okay, I'm trying to remember. I know it's discussed in this particular module. So before I move off this slide, so all your genetic information, that is the library of information that's transferred from one cell to the next, to the daughter cells, or parent to daughter cells, is found in the genome. Now the genome is for a prokaryotic cell is going to be that it's generally circular and usually just one copy, so haploid, and is found in the nucleoid, right? Some bacterial cells will also have what we call plasmids. Plasmids are extra chromosomal DNA. So they're DNA that's not associated with the chromosome itself. And they will usually have an autonomous process for DNA replication. So they're its own structure and it replicates. And so that, for a bacterial cell, that is the source of the genetic information. The chromosome itself, and then potentially, if it has a plasmid, the information in the plasmid. A plasmid usually confers extra information that's not required for the cell. as far as cell growth is concerned, but it provides a benefit of some sort. Things such as, so our plasmids provide, say, antibiotic resistance. So it's a resistance plasmid. The F plasmid provides the ability of the cell, that particular organism to produce the sex pili. Then we have other plasmids that might give it the ability to additional virulence factors, ability to establish in the host as a disease-causing organism, where maybe without that plasmid they can grow, but they don't establish as well, so they're less likely to cause disease. But with the plasmid, it provides them information to produce these enzymes or toxins that really help it establish the disease state. For a eukaryotic cell, okay, the eukaryotic cell, the genome is going to be found in the nucleus. It is linear in structure. It is diploid, so you have 2 copies of each. Let's see, we also find, so it's in the nucleus, we also can find that part of the genome in the mitochondria or the chloroplast if the organism has those particular structures, okay? So if whichever structures of those 2 that they have, that DNA that's found in those organelles is part of the genome. Some of your fungi and single-celled or other single-celled organisms like the proteins might also have plasmids, okay, as another source for that genome. All right. Now, as far as DNA replication is concerned, so this textbook is going to talk to you, it's just going to kind of go into the whole how they discovered the process for replication and the fact that it is semi-conservative, right? They just talk about the 3 different options that were out there. I'm not so interested, I mean, it's good, well, as a science person, I think it's good reading material. If you're not as, thrilled about science, that aspect of science, it might not be of interest to you. I'm not going to be testing you on those 3 different, and the process they got to in order to get to semi-conservative. What you're really walking away with is that it is a semi-conservative process. So the parent cell has, its DNA is double-stranded, right? And so, during DNA replication, those 2 strands are going to be separated, and each strand will serve as a template for a complementary strand to produce then the 2 new DNA molecules. And the 2 new DNA molecules have one of the parental and one new strand, okay? So that would be the first cell division, with the first cell division. And so in the second generation, this, black pen. There you go. We won't lose either, at this point, either parental. Now we have a parental that is red, okay? We have a parental that is blue. Okay, combination. And then these daughter cells will now have the new strand. Right? So, second generation, we still have the light blue. Okay? The first generation, we still have the blue. The second generation, we still have the blue. As we go to the 3rd generation, the 4th generation, the 5th generation, that original parental stays in one of the daughter cells of the next generations. It doesn't go anywhere. It is always going to remain. That's that concept of semi-conservative. Each cell conserves half of that genetic information from the original with a new strand to create the new molecule. Okay? All right. Now, as far as DNA replication itself is concerned, DNA replication consists of 3 steps: initiation, elongation, and termination. Okay? Initiation is going to mean that we need to recognize the origin of replication. In bacterial cells with a circular DNA, right, there is only going to be one origin of replication. That is that one place needs to be recognized, and from there we will start DNA replication. Now we are going to stick to very basic, simplistic process of the DNA replication. However, I will make a comment. It's the same level you had with general biology, okay, which is, again, very basic. We are not going into, this is much more involved than what you actually have gone through in general biology or even this class. They have a class, molecular biology, that's going to go into far more detail. There's a lot more proteins and enzymes involved in the process than what you guys have probably talked about so far in your education. And we're going to keep it that way. I just like to point out, though, that we're keeping it basic. It's actually much more complex than what we even talk about. Okay? And so it's good to recognize that there is more going on in the background to make this whole process work. But we're going to stick with the big players, okay, instead of all of the supporting team. Okay, but at the origin of replication, we're going to need some of these proteins recognizing the origin of replication. The key is to get helicase at this location. Helicase is going to, when it binds, it's going to break those bonds between the 2 DNA molecules and separate them into individual strands. That's its job. And in doing so, it creates what we call the replication bubble. Now, the replication bubble is going to have a fork on either side. The fork is where so the 2 DNA molecules come back together. I'm not going to try to do the double helix aspect of this structure. Okay, so the replication fork is what's being pointed at right there. Okay, the replication fork is where they act during DNA replication, and that's where the bulk of the activity is going to be happening, because this is where, as the process continues, this is where helicase will remain at this location and keep separating out the DNA strands. Okay. In that process, though, of course, we're already going to have new strands created, but for a short period of time, the DNA is going, the 2 DNA strands being separated are going to be single-stranded. During that time, to protect that DNA and prevent them from coming back together and base pairing back together, we have our single-stranded binding proteins. So they'll be right around that, they'll be right around the replication fork and anywhere where the DNA molecule has been separated into individual strands. Okay? Because DNA is going to have a tendency to come back together automatically on its own. Now, once we've got our 2 strands separated, we are going to have the enzyme primase come involved. And primase is an RNA polymerase. And what it's going to do is lay down a short segment of RNA. You say, well, wait, I thought we were doing DNA replication. And we are. Here's the thing. Our big players in DNA replication and actually polymerizing the reaction are DNA polymerase, the 2 would be DNA polymerase 3 and DNA polymerase 1. I don't know why I must have hit that. That should not be an exclamation mark. It should be a 1. But those are the 2 that are big players, and they have one fault to them. Okay, and their one fault, well, they have multiple faults, but one of their faults is that they cannot take a nucleotide and a single nucleotide and another single nucleotide and attach them. Okay, they don't have that capability. They can only take a nucleotide and attach it to a growing strand, a strand that already exists. Okay, they can't take in 2 individuals, like 2 little Legos. They can't take 2 little Legos and attach them together. But if you already have a Lego structure going, they can attach it to that. Okay, that is one of their downfalls. And therefore, the role of primase is to create that little length that they can add to, that polymer that they can add to, okay? So primase is going to lay down that primer. It is an RNA primer, so it will be removed at some point and replaced, okay? It doesn't stay there permanently, it's just a temporary hold in order for RNA polymerase to add on. In addition, there is something called DNA gyrase. DNA gyrase has multiple functions. First is that it's going to help when you have two strands of DNA and they're twisted, double helix, so they're twisted. When you try to separate them, if it's linear, you don't have this problem as much, but when it's circular, what's going to happen is you're going to get what we call supercoiling. Okay, as you separate that portion, this portion right here, it's going to tighten the coils around the rest of that structure. So the DNA gyrase its job is to go in there and break the strand DNA and release that supercoiling. Okay, so that DNA replication can proceed, and it'll continue to do that throughout the the process of breaking and resealing that DNA to release the supercoiling. Now, when it comes to elongation, I have this next slide is going to be actually looking at elongation step, and that will be, so we have our RNA primer laid down. Now we move into elongation. For elongation, we have our leading strand and our lagging strand. They get their name because the leading strand is one continuous strand that's being produced, okay? 5' to 3', because we're always going 5' to 3', and it's moving towards the replication fork. The lagging strand is slightly behind, okay? And it has the behind aspect is because it is going away, the net movement and production is towards the replication fork, but the actual movement for DNA polymerase III to make its complementary copy is away from the replication fork. And so what it does is creates these little fragments called Okazaki fragments. And then we have DNA polymerase I that comes in and removes the primer and replaces it, right? And Okazaki fragments are then linked by ligase, okay? Because again, another downfall of our DNA polymerases is they can't take two segments of DNA and attach them, okay? They can only add a nucleotide to a growing strand. So ligase is able to take two segments, two fragments, and attach to make a single. Now, because of these Okazaki fragments, we're going to have primase is going to play a major role in elongation for the lagging strand, because it will need to add in a primer every 100 or so positions in this process. And then lastly is the termination. Termination is going to be when replication reaches the other side, and then we need to separate out these two new DNA molecules, okay? For that, we have topoisomerase 4 that is going to disconnect the two structures so that we now have two separate DNA molecules. The DNA replication process is going to occur bidirectionally along that circular DNA. So it's a circular piece of DNA. Replication is going to occur both directions until they meet on the other side. Okay, so the origin of replication is on one side, termination point is on the opposite side. So what we have for your leading and lagging strands, yeah, I'm debating here whether, I don't know where my breaking point is going to be here. Try and get through this. All right, so this picture right here is demonstrating, say for instance, this right here, okay? That's what you're seeing. And so you're only seeing, half of the replication bubble. Keep in mind that the other half is doing the exact same thing. This process occurs bidirectionally, okay? The top is showing you what we refer to as the leading strand. So it is where that DNA polymerase III is going to be going 5' to 3' towards the replication fork. As helicase unwinds the DNA, new template is provided, DNA polymerase III can continue on in and fill in and create that complementary strand, okay? Therefore, it only needs primase one time at that origin of replication. From then, it's just moving on in to that replication fork as that template is being provided. It's a continuous process. Where all of the excitement and kind of the messiness that's happening is over here in the lagging strand. For the lagging strand, this template now, because don't forget our two DNA molecules are anti-parallel. So for this template now, to go to polymerize 5' to 3', you can't go in this direction to the left. You have to go this direction to the right, okay? And there's no way that the cell can sustain keeping that template open with single-stranded binding proteins for an extended period of time. So it needs to use that template immediately to make a complementary copy, get our double-stranded going, and repeat in that situation. And so what's going to happen is, as template is provided, the replication fork opens up, and new DNA is provided. Primase is going to lay down a primer. DNA polymerase is going to extend off of that primer until it meets the previous location, the previous Okazaki fragment, okay, the previous primer that was laid. So it butts up against that, okay? DNA polymerase then can, at that point, relocate to the new primer that was laid down and go. So it's making these little segments, okay, those Okazaki fragments, and now we need those Okazaki fragments to be attached. That is the job of DNA polymerase I and DNA ligase. DNA polymerase I is going to come in. Notice our RNA primer is in green. Okay. Notice DNA polymerase has the has the ability to remove the RNA and then extend off of that Okazaki fragment until eventually it butts up with the next Okazaki fragment. DNA polymerase I can't make that connection between this fragment and this growing strand. So that's where DNA ligase comes into play, and it makes the final connection. Okay? So although we're making short segments and we're going away from the replication fork, in the end, the net production is still getting bigger and bigger in this direction. Does that make sense? So that we are making a longer and longer product, that DNA, new strand of DNA, that's a template to that parental DNA that's being supplied as the replication fork is moving along that DNA strand. Okay, many more players involved in the lagging strand. It's always a little bit behind because there's a lot of extra steps involved. One thing here, well, two. Once the primer is laid down, DNA polymerase, and that's it. Then that's it. That's what DNA, the RNA primer laid down by primase, followed by DNA polymerase III, and it just keeps going, 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 right? Like Energizer Bunny until it reaches the other side. Versus what we have over here is short segments. Okay. I will stress on this next picture is just stressing that idea of the circular DNA. When DNA replication occurs, the circular DNA is going to go bidirectionally Okay, so we now have the two strands are separating here. At this point, our replication forks are further down that circular DNA, but eventually they're going to meet up on the other side. That's the termination side. That's where topoisomerase 4 is going to come into play to separate. Okay, this is also where you can kind of get an idea. If I start pulling these, okay, away from each other, what's going to happen here? It's going to get tighter twisting. That's the supercoiling the DNA gyrase is going to be releasing. Okay, because it's a single strand of, it's a circular strand of DNA. Take some yarn at home, tie it to a knot so you have a circular piece of yarn, right? And then try to separate out the strands of the yarn. You're gonna be able to get them separated initially, and then you're gonna reach a certain point where you won't be able to move any further because the supercoiling will have will be too tight, right, for you to proceed to separate any further. So that is one of the problems that has to be addressed with circular DNA, and that's the job of DNA gyrase, is to release that. As compared to eukaryotic DNA is